Genotyping by sequencing method. Illumina sequencing. Genotyping by sequencing method. GBS method to discover single nucleotide polymorphisms in order to perform genotyping studies such as genome-wide association studies. GBS simple, highly multiplexed system for constructing libraries for next generation sequencing. Its advantages are reduced sample handling, few PCR and purification steps, no DNA size fractionation, efficient barcoding system. The steps involved in GBS method are quantify genomic DNA, normalize, add restriction enzyme, then add adapter and ligase, and pool samples, then PCR, clean and size library, then sequences are converted into FASTQ file. Illumina sequencing method. Illumina sequencing is a technique used to determine the series of base pairs in DNA, also known as DNA sequencing. The Illumina machine. The steps involved in Illumina sequencing are: first, the library construction. Fragment, attach adapter DNA. Step 2, cluster generation. Add to flow cell, then bridge amplification. Step 3, sequencing. Single base at a time, imaging. Step 4, data analysis. Images transformed into base calls and reads. Procedures involved in Illumina sequencing are tagmentation, reduced cycle amplification, bridge amplification, clonal amplification, sequence by synthesis, data analysis. Procedure 1, tagmentation. First step after DNA purification is tagmentation. Transpostasis enzyme randomly cut the DNA into short segments, then adapters are added. Procedure 2, reduced cycle amplification. During this step, sequences for primer binding, indices and terminal sequences are added. Indices are usually 6 base pairs long and are used during DNA sequence analysis to identify samples. The terminal sequences are used for attaching the DNA strand to the flow cell. The oligos match the two kinds of terminal sequences added to the DNA. Procedure 3, bridge amplification. It involves cluster generation and creating hundreds of DNA identical strands, and then create forward strand and a reverse strand. Polymerases move along a strand of DNA, creating its complementary strand. The original strand is washed away, leaving only the reverse strand. At the top of the reverse strand, there is an adapter sequence. The DNA strand bends and attaches to the oligo that is complementary to the top adapter sequence. Polymerases attach to the reverse strand of its complementary strand. The illustration shows how tagmentation, reduced cycle amplification and bridge amplification occur. This illustration shows the clonal amplification. Here, the DNA folds over into a bridge, like shaped. Then add the primer. The primer makes the complementary strand and separate the reverse and forward strand. Then clonal copies of both forward and reverse strand in a cluster. Again, DNA strands will bend and attach to oligos. Polymerases will synthesize a new strand to create a double strand segment, and that will be denatured. All of the DNA strands in one area are from a single source. Clonal amplification is important for quality control purposes. Illumina sequencing uses polymerases based substitution. Errors have been observed, especially at the 3' end. Sequence by synthesis. All of the reverse strands are washed off, leaving only the forward strands. Primers attach to the forward strands, and a polymerase adds fluorescently tagged nucleotides to the DNA strand. The polymerase sequences the complementary strand on the top of the strand. They separate and the 3' end of each strand is blocked. The forward strand is washed away and the process of sequence by synthesis repeats for the reverse strand. Procedure 4. Data analysis. The sequencing occurs for millions of clusters at once, and each cluster has 1000 identical copies of a DNA insert. The sequence data is analyzed by finding fragments with overlapping areas. High sequencing can produce read lengths around 90 base pairs long. This illustration shows the clonal amplification, sequence by synthesis, and data analysis. Advantages of Illumina sequencing: to sequence multiple strands at once and gain actual sequencing data quickly. Then, the uses of Illumina sequencing are whole genome and region sequencing, transcriptome analysis, metagenomics, small RNA discovery, methylation profiling, genome-wide protein, nucleic acid interaction analysis. Illumina sequencing has been used to research transcriptomes of the sweet potato and the gymnosperm genus.